And there we go. So yeah, we're at a nice part of the course now, midway through solution properties of antibodies. And this follows on nicely from Roy Jeffress's lecture last week, because he talked about aggregation and the formation of these complexes, which can then also become very large between antibody and antigen. But how do we quantify the presence of aggregates? Because aggregates can be immunogenic, they can cause problems. So it's very important when making formulations of antibodies, monoclonal formulations, or antibody fragments formulations, that we make sure that the materials have full molecular integrity, and they haven't formed aggregates or indeed fallen apart. So we want to now look at the methods, or the principal method, one of the principal methods used to establish if the antibodies are aggregated or not, and also to check on the conformation of the antibodies. It's also particularly important when dealing with the high concentration formulations, which are used for monoclonal antibody delivery, which can be in excess of 100 milligram per mil. That's 10%. That's very high. So knowledge of the solution properties of antibodies is important, and that's what we're going to be talking about today. So if I just come out of that and then switch to PowerPoint, let's just see if we can just capture that. Okay, folks, can you see that? It should say solution properties of antibodies.
Yeah, we can see it.
Brilliant. Okay, so for the next hour or so, and we'll see how it goes. If it's going on a long time, we might have a break in between all this. So it's about how we quantify the molecular integrity of antibodies, the presence or not of aggregates, and whether the molecules are intact or not. So purity, including presence or not of aggregates, and conformation. Purity in terms of these distribution profiles. So this one here will be a very impure antibody preparation because of the presence of multiple components presumed to be aggregates. So we're going to focus on purity, aggregation, and in terms of conformation of antibody molecules, which, although we see in textbooks, are these Y and T shapes, in reality they're not necessarily T or Y shapes, but are three-dimensional entities where the FC and the two fab fragments can be in different planes. And how we represent the conformations of antibodies in in solution. Antibodies can be very difficult to establish conformations, structures. Techniques like X-ray crystallography can struggle in terms of trying to produce structures, because antibodies are, or most of them anyway, are intrinsically flexible structures. Does anybody know which part of the antibody molecule which confers this flexibility?
I think the hinge region.
Absolutely, the hinge region. So the hinge region confers flexibility. And of course, some antibody classes or subclasses have a significant hinge region. IgG3, for example, has a very long hinge region, whereas some antibody classes or subclasses have very little in terms of hinge properties. Did anybody name an antibody which has an absence or almost a complete absence of a hinge region?
I think IgG1.
That's got a short hinge region, but still significant, as IgG2, IgG4. But this model here, called a Bead model, is of a particular class of antibody which doesn't have much of a hinge region. In fact, it has an extra constant domain. This is the model for IgE. So IgE is a class of antibody molecule which doesn't have significant flexibility because of the extra constant domain region rather than the hinge region. So we were talking about how we assess the purity of antibody molecules or otherwise, and how we can get an idea of the conformations of antibodies in solution. And to get round this perception that antibodies are T or Y-shaped structures, which is useful in terms of a 2D representation, but doesn't reflect what the conformations are in nature. And the technique we're going to focus on is this technique or instrument called an analytical ultracentrifuge. Okay, first of all, it's an instrument, it's not a machine. And a machine is what is like a tool which you press buttons and it does operations and that's it. Whereas a scientific instrument is one that you have to interact with to get the required measurement. Like a musical instrument, you have to interact with a musical instrument in order to get music out, as opposed to a stereo hi-fi system, which you just press a button and you get the music out that way. So a stereo hi-fi system is a machine, and a violin or a piano is an instrument. The same with scientific instruments that you have to interact with and understand in order to get high quality experimental measurements. And that's what an analytical ultracentrifuge is, which I know it looks like a washing machine, but this instrument, which is the Optima ultracentrifuge, analytical centrifuge, is regarded as a gold standard instrument for assessing the solution state of antibody molecules. So it's worthwhile focusing on this one as part of our antibody biotechnology course. Now, the MRes students, you guys have already had an introduction to this technique as part of your other modular training, but the biotechnology students have not had this yet. So am I correct in assuming, this is talking to the MSc Biotech students now, that you've not met this type of instrumentation before? Is that right? You may have met techniques like electrophoresis or chromatography, but you've not met instrumentation like this before. Is that a correct assumption, folks?
For me, I have met the small one, centrifugation, not the ultracentrifugation, which require high speed of the RPMs.
Who's that speaking?
Khalil.
Khalil, yeah, but you've also had a lecture on this last semester with but I don't think the biotech students had that either. Yeah, but I think as Khalil said, I think many of you have met sort of small centrifuges where you're spinning materials to, clarify a preparation from cell debris and all this sort of stuff. So I think some of you, many of you might have met bench type centrifuges before, but maybe not this type of instrumentation here, which has an optical system. So what I'm going to do now, I'm going to show you a couple of slides giving the basic principles of the technique, and then there are a couple of short videos. I don't know whether you got my announcement either this morning or yesterday, but it was a request that if you had the chance to look at two short video clips. One lasted about a minute and a half, introducing the technique, and the second one was a broadcast on the local NOTS TV, where the centrifuge was being used to characterize not antibody complexes, but complexes of an antibiotic, vancomycin, with mucus, with mucin glycoproteins. Did anybody have a chance to see those clips?
Yes, me, Steve.
Okay, well, we can show them briefly again. So we're going to show the first 2 or 3 slides introducing the method, and then we'll show the clips, and then we'll then go through and then show you how this technology, this instrumentation is used to quantify purity, aggregation, and conformation. Not in great depth, but sufficient for you to understand how this important technique can give this sort of information that Roy Jeffress was talking about last week. There's no point telling you that antibodies can form aggregates if you don't know then how to establish whether you've got aggregates or not. Right, so this is the ultracentrifuge. It's called an ultracentrifuge because it's high speed. Okay, high speed means it can get to speeds of 60,000 revolutions per minute. So that's the ultracentrifuge. It's an analytical ultracentrifuge because it's got doesn't just spin things at very high speed, spin your solutions of your protein or antibody or antibiotics or whatever at very high speed, 60,000 revolutions per minute, where you can get centrifugal fields of 100,000, 200,000 G. So it's like being on the surface of of Jupiter or something. It's the gravitational fields are very, very high, which is why you can sediment small particles like proteins, DNA, carbohydrates, and things. But it's an analytical one because it's got a special optical system which can actually let you follow the sedimentation process under very high speeds. And this actually is an optical pattern on the computer here, which comes from the optical system that's built into the computer. This actually is an optical pattern based on ultraviolet absorption characteristics of a protein molecule. It's actually part of a sedimentation equilibrium experiment. But it's one of two types of optical system which are on modern instrumentation. And what you have are essentially, I think maybe what we'll do. No, I'll show the clips now. Maybe that's the best thing. Let's show these two clips now. So what I'm going to do, as I always do, let's come out of the slide presentation. Right. So if you guys open your meeting chat, folks, you got meeting chat opened, and what I'm going to do is just go to right. Let's just get these links again. Copy, put that in. Right, let's just put that in that file there. Notepad. Goes in like that. And then I'll put the second clip on as well. And that would have been right. Let's go to that again. Just bear with me 1 second, folks. Playlists. Go to Biophysics. Copy link address. Put that there. Okay, let's just copy that, and then I'll put it in the chat box. Right, folks, so I've just sent you 2 links, 2 video links. Can you all see that? So when I give the signal, if you can click on the first link, that's what it says, lecture notes, analytical ultracentrifugation clip.mpg, and click that. Before I do so, just warn you that this recording was made from an American laboratory, and at a time when health and safety regulations weren't like they are now. So I think one of the guys is not wearing a laboratory coat. Okay, practice doesn't happen anymore. So we were laboratory coats now in doing this type of measurement. So let's just play this clip through. So if I 1, 2, 3, if you can click that link, and then it lasts about 2 minutes, and we'll reconvene back here in 5 minutes time. Okay, 1, 2, 3, click. Ready? Go. Hello. Did you see that clip?
Yes, we did.
Okay. Right. What I'd like to do now is click on the. So what you saw there was basically a scientist loading a rotor into a centrifuge, and inside that rotor it had 8 holes, and in those 8 holes were special cells. And in the second clip you should see what those cells are like. I don't mean human cells, I mean containers which contain the solution that you are analyzing. In this case it'll be the antibody solution, and there's 2 sectors. One has the solution of your antibody or whatever it is, and the other has a reference solvent, which is the solvent which your antibody is dissolved in. So you've got reference and you've got your solution, and you put it inside this rotor chamber, and then the chamber is closed off, and then it's spun at very, very high speeds. But because it's high speed, you've got to have a vacuum. So you've got to have a very high vacuum. Otherwise, if this thing is spinning at high speed in air, then things get very, very, very hot because of the air resistance. So at these high speeds, you've got to have a vacuum. So that's what was happening at the start. He was putting the rotor with the samples inside this rotor chamber, closing it off, switching on the vacuum, and then doing some measurements to follow the sedimentation of the molecules. And that clip showed that at high speeds, the bigger particles tend to sediment faster compared to the smaller ones. So the bigger ones tend to move faster. So if you've got any aggregates in your solution, they will sediment at a faster rate compared to the smaller ones. And also, it depends on the conformation, because a compact blob-like molecule, when it's sedimenting through a solution under the influence of a very high centrifugal field, a small compact molecule will experience less frictional resistance going through compared to something which is like that, which will experience a lot of resistance. So the rate at which the molecule, or whatever it is, particle sediments, depends on how compact it is, because the more compact, the faster it goes, and also how big the particle is. The bigger the particle is, the higher the molecular weight, the faster it will go. So this technique is a measure of the mass and on the conformation, how compact or not the molecule is. So this second clip, which is taken from the local TV was broadcast about a year ago. It's got Dr. Mary Phillips-Jones in, who is in the NCMH, and the IPB students will be meeting her later on when we do the polysaccharide biotechnology module. But the biotechnology group and you'll be interested in what she does because she works in the field of antimicrobial resistance, and she looks at antibiotics like vancomycin and things, and how they act on receptors on bacterial cell surfaces, and the clever way that the bacteria can react to these antibiotics, a bit like the COVID situation with the how clever I say clever, COVID doesn't have a brain, but the way that it reacts to defend itself is unfortunately from our point of view very clever in developing resistant strains. So she's very much involved with that. And this particular clip is some work we've been doing on the oral administration of vancomycin and how it causes mucin, which lines our alimentary tracts, to clump together. Whether that's a good thing or bad thing or not in terms of delivery of the antibiotic is another question. So on the next clip, again, don't focus so much on the microbiology bit, I'm afraid. Just focus on the ultracentrifuge, which is being used to characterize the size of the complexes, and in particular focus on the containers which are used to put the what you put the solutions in, how they're loaded into the rotor, and see how the rotor is put in the instrument. And the researchers are wearing laboratory coats. So Dr. Phillips Jones, myself, thank goodness, and also Dr. Vlad Dinu, who's doing the assemblies. We're all wearing laboratory coats, and it's quite a nice piece. It's only about, again, 3 minutes. So I'm going to get the signal, folks. So if we do the 1, 2, 3, can you click and see how we go? Ready? 1, 2, 3, click the second piece. Okay, folks, have we all seen those clips? Great. And yeah, thanks, Anastasia. I think not to do with the antibodies, but there's an important point raised, is that Mary talks about in the talk about not disposing irresponsibly of medicines, and what you should do really is take them back to your pharmacy store, and they will dispose of them properly. With antibiotics in particular, you don't put them down the toilet or the sink. That's not a good idea. But return them to your local pharmacy store. Hope that's clear. Yeah, so good point. Thanks for raising that. Okay, folks. So if we're all back on board, I'll just go back to those slides again. All right, folks, are you with me again? Have you all got the PowerPoint presentation? Yep.
Yeah.
Okay, so that's the the instrument, the analytical ultracentrifuge, and these are the containers, cells, not plastic tubes you use in a preparative centrifuge, but this is an analytical one, which we're analyzing the sedimentation process. So you use these special containers called cells, which are quite expensive. So they cost about over £1,000 each. So at the end of an experiment, you don't throw them in the bin. These are reused time and time again over a period of many years, and contain many, several high precision components in them, which are kept scrupulously clean. See the two sectors here? These, when this is assembled together, you put the solution in one sector and the reference solvent in the other. So that's the reference solvent in the left and the solution in the right after everything has been assembled. And these are transparent windows here which go at the end. So that means when the cell is fully assembled, you can pass light through them of the appropriate optical properties. So you can actually record the sedimentation process as the solution is spinning at very high speed. So you spin like this, and the centrifugal field goes radially outwards away from the center of the rotor. There we are. This is you see that this is the cell looked end on. See those two little channels? This, by the way, is a poor substitution. What we would have done in past years is before or after the lecture, I would have taken you over to the NCMH laboratories and show you this instrumentation in action. So showing you the TV clip and these clips from these snapshots from that clip is very much a poor substitute. And maybe when the lockdown restrictions ease off a bit, which hopefully will be quite soon from what the government said yesterday, we'll be able to show you the operation of instrumentation like this. So maybe in a few weeks' time, I'll be back in contact with you over that. And then you put your cell, when it's loaded up, into a rotor, and notice there's several holes in the rotor. Any centrifuge has to be balanced, so the weight this side has got to be balanced by an equal weight the other side, and you can balance it with another cell which has got a similar amount of material in, and there's room for more as well. But you always have in one of the positions a what's called a counterpoise cell, which helps you to calibrate what you see on the computer in terms of actual distances from the center of the rotation, which is the center of this spindle locator here. So very high speeds, up to 60,000 revolutions per minute. And when it's spinning this way, you get very large centrifugal fields coming out from the center, as much as 200,000 G. But these high G forces, very small particles like proteins, antibodies, and other types of molecules will sediment. Okay, right. So can you all see that still? Can you see this slide now where I'm showing you, it's a schematic. Can you see this drawing?
Yes, we can see it.
So if we're looking down on one of these cells and they have these two compartments, one contains pure solvent, but the other, which is the reference, the other contains your solution. So you're looking down on one of these sectors. Then after a period of time under the influence of a high centrifugal field, what happens is that the protein sediments towards the outer radial part of the cell. So it sediments in an outward fashion until it reaches the bottom of the cell. But this can take, a period of an hour, 2 hours, 3 hours to happen. And what happens is that as the stuff slowly sediments, it leaves a region of clear solvent left behind as the protein moves towards the bottom of the cell. So if we were to scan across this cell for the presence of protein, and we can do that by using, for example, ultraviolet absorption optics, because proteins, or most proteins, absorb in the ultraviolet region around 280 nanometers because of all the aromatic groups in the protein side chains like tryptophan, tyrosine, and they absorb UV radiation at 280 nanometers. But we can also use a different type of optics called refractometric or interference optics, which will give you the same trace. So if we scanned across here with our refractometric optics or our UV absorption optics, we will find that this region here has very low absorption because there's no protein there. And then you have this region has high absorption of UV radiation because that's where the protein is. And then in between, you've got this boundary between solvent and solution. And the rate of movement or rate of change of this sedimentation profile gives you the sedimentation coefficient. And the sedimentation coefficient is simply the sedimentation rate divided by the centrifugal field. And the units of the sedimentation coefficient are Svedbergs, and 1 Svedberg is 10 to the minus 13 seconds. And you will have heard of this unit because when we're describing, for example, the ribosomes, you know, the 70S, 30S ribosomes, the S refers to the sedimentation coefficient in Svedberg units. And the larger the S value, the faster it sediments, and generally speaking, the bigger the molecule. So the 70S ribosome will be bigger than the 30S ribosome. And in food science, you've got the 12S, 11S, C globulins, all this sort of stuff. So it's used as a measure of size. The sedimentation coefficient, symbol S. What this means is there's a correction for what we call non-ideal effects. And this means that we've standardized the comparisons to water as solvent at a temperature of 20 degrees centigrade. So the special formula which allow us to convert our sedimentation coefficient measured in a particular buffer or in glycerol or something, and convert it to standard conditions, which is the density and viscosity of water at 20 degrees centigrade. So typically an IgG antibody would have a sedimentation coefficient of between 6 and 7 Svedbergs. There's also a second type of sedimentation experiment in which we don't sediment quite at high speed, so we sediment at lower speed. You don't sediment at 60,000 RPM, but we say we sediment at 10,000 RPM, which is still quite fast. And at these lower centrifugal fields, what happens is that the sedimentation forces, which are trying to sediment everything to the bottom of the cell, there's something else which doesn't like that. It wants everything to be pushed back again. And this force backwards is called diffusion. So centrifugal force is this way, and diffusion tries to push everything back. Now, at very high speeds, the diffusion backwards force is quite small compared to the centrifugal force. So at high speeds, the centrifugal force wins and you get sedimentation. But at lower speeds, say at 10, 20,000 revolutions per minute, these forces become comparable. and you don't get sedimentation towards the bottom, but instead you get a balance or equilibrium between the sedimentation force in this direction and diffusion force going backwards, and you end up not with a boundary which is moving towards the bottom of the cell, but you end up with a steady state pattern of a low concentration at the solvent meniscus, gradually building up to a higher concentration at the base. And when these forces come to balance, equilibrium, this pattern stays the same. It doesn't move. It's that equilibrium. And because it doesn't move, then remember I said that the sedimentation rate depends on the size and also how compact the particle is. With if it's not so compact, it'll experience frictional forces and get slowed down. Well, in sedimentation equilibrium, because there's no movement when we've got equilibrium, then there's no friction forces. So this pattern is purely a function, whoops, what have I done? Is purely a function of the molecular weight and not of the conformation. Whereas the sedimentation coefficient is a measure of the molecular weight, the size, and the conformation, because the sedimentation rate depends on both. So with the same instrumentation, a centrifuge, we can get two types of measurement. Sedimentation velocity, where we measure the sedimentation coefficient, the Svedberg value, and at slower speeds, we can measure the molecular weight or molecular weight distribution with the sedimentation equilibrium technique. So it's a very versatile instrument, and that's one of the reasons why the FDA, the Food Drug Administration, like this type of instrumentation. And as I said, particularly the biotech students, you don't need to know the physics between how this is all done. You just need to know that this technique can be used to measure the sedimentation rate and the size and conformation with sedimentation velocity and molecular weight with this technique. But also it can be used to assess the purity of a molecule. Because all this information is going into a computer, we can not only find the average sedimentation coefficient of all the materials inside your solution, we can get a distribution of sedimentation coefficients. So if you've got different things present, like different proteins or maybe aggregates present, then this technique will tell us, be able to interpret how this concentration distribution changes with time in terms of the sedimentation coefficient distribution or heterogeneity of a preparation. And this is the first use of this instrumentation for antibodies, is it can be used to assess the purity. We can use sedimentation velocity in the centrifuge and obtain distributions of the sedimentation coefficient. And this is one such distribution. This is obtained, this is a distribution of sedimentation coefficients. So you've got sedimentation coefficients ranging from about 5 Svedbergs all the way through to about 15 Svedbergs. So this would not be a pure preparation. A pure preparation would have everything that's around, for an antibody, around 6.7 Svedbergs. So we've got a peak around here, 6.7. We've got another peak here, 9.86. Another peak here, 12.74. And another peak here at 14.71. So you've got this heterogeneous mixture of components. And I won't tell you which company this came from, but one of those companies, well, they're a company that are associated with the vaccine that I received at the Queen's Medical Centre last week. I can't say any more than that, although I might get myself into trouble. But this preparation is an antibody from that particular company, and they sent it to us as part of a PhD studentship of someone who did the MSc with us in applied biotechnology, and then went on to do a PhD with this particular company, paid by this company. And they were interested in the effects of storage, freeze store and long-term storage on the stability of antibodies, and wanted to know how long an antibody would be kept, or how many freeze store cycles that you could do before you ended up with aggregation products. And this is a very aggregated sample. So this is the main peak here, which corresponds to the pure antibody, which has a sedimentation coefficient of 6.7 Svedbergs, and there's 74.9% of this. This is dimer, which is 9.86 Svedbergs. There was no saying, I think. Was it something like dimers are the girl's best friend? Is that right? Or was it something else? Was it diamonds are the girl's best friend? Not sure. But dimers certainly were not the best friend of this this company, because this represents aggregate. And then you have another set of aggregates here. There's 18.8% of this dimer. There's 4.9% of this material here, which is 12.74 Svedbergs, and there's 1.4% of this 14.74 Svedberg aggregate. So this is a very impure preparation. So what the company would be trying to do would be to find some form of a stable preparation where there was only one monomer peak. So this value would be 99.9%. And this is the sort of thing which a company would have to present to the FDA, the Food Drug Administration, to show that they had pure antibodies which could be injected into a patient. So if I was a patient, I wouldn't want that preparation injected into me. And this is even worse preparation. Look at this. This is IgG4 again, and we've got an even lower amount of the monomer, only 52.3%, and 25% dimer. That's bad. And also even stuff that's very high, 25.9 Svedbergs. So this would not be a very good preparation and would fail the criteria that the industry would have in terms of what can be injected into a patient. But it's very important, and as I said before, it's particularly important with monoclonal preparations, which are used at very high concentration. So there's the antibody preparations that Roy Jeffress was talking about last week, including rituximab, they will be required at very high concentrations, in which at high concentrations have a higher risk of aggregation processes. And this method is a very important check on aggregation. And the reason why the industry likes it is that there are alternative methods for getting aggregation. There's a technique known as gel chromatography or gel filtration chromatography, which involves putting your solution down a gel permeation or size exclusion chromatography column. And that will separate on the basis of size. So the larger molecules would come off first, so you'd be able to see if there was aggregates present. But unfortunately, there's always the worry with column chromatography of your material interacting with the materials of the column. We call this a matrix problem, whereas the ultracentrifuge, the separation is based purely on size and how that size reacts to a gravitational field. So there's no need for a separation matrix at all, and therefore no problems in terms of assuming inertness. So that's why the analytical ultracentrifuge is deemed as a gold standard method. And also, if parts of the antibody molecule were coming apart, like the Fag fragment or FC, you'll be able to pick that up as a peak at a lower value of the sedimentation coefficient. So this would fail quality control. As I said, what you'd be looking for is preparations with 99.9% monomer purity. So depending on what sort of antibody you have, IgE or IgG3, IG1, IG2, typically sedimentation coefficients are around 6 to 7 Svedberg units. So smaller than the ribosome. Right, so any questions on that? That's how we use the centrifuge to assess the purity and the presence of aggregates or not. So as you can see, I'm not at Sutton Bonnet, I'm afraid. We're still locked down following the rules, but from what the government said yesterday, it looks rather promising now in terms of return to normality, and that includes practical work as well. So that's a very good sign. So in the background there, you can see this is my back room. That's a piano, okay? And the piano, is that an instrument or a machine? It's an instrument. That's right. So the same way I said before with the ultracentrifuge or scientific equipment, always regard them as instruments rather than the machines. So they're even though they may be in a box with stuff all contained in there, they're still scientific instruments, and then you have to interact with the instrumentation and understand how it operates, not obviously the detailed physics, but understand the generation of the measurements and be able to judge whether a measurement coming out appears accurate or reliable or not. And be prepared to test an instrument out by putting a standard material down there and see if it gets the approach it's a good thing, and checking it standard materials to correctly. And that's certainly true with the ultracentrifuge. Making sure the temperature measurement system is going, that the speed is correct. What it says on the dial is exactly what the speed is. So if these things are not right, then that can give erroneous data and make you think something is when it isn't, and vice versa. Okay, let's go back to the so for the second part, that's pure. We just dealt with purity and how we assess for the presence of aggregates using the ultracentrifuge. The second thing is in checking on the conformation of the molecule, the antibody molecule, and they're quite unusual molecules. They're quite because they have these three main domains, an FC and two Fab domains, linked together usually by a changing the glycoform or pegylating the antibodies, which is what not only to solve but also okay.
If sorry, we can't hear you. The connection was interrupted frequently.
Can you hear me now, folks? Hello, can you hear me?
Sorry, Steve, we haven't heard you perfect because the connection was interrupted.
Right, I can hear you okay. Let's just come out of the recording. Let's come out of the screen. Okay, folks, am I still breaking up?
Yes.
Oh dear. I mean, one solution is to have a longer break and then see if I can fix the problem. What's happening at 4 o'clock, folks? Have you guys got another lecture at 4 o'clock? Ask the question by dear. Okay. Let me change the check the connection at the back here. Okay, I've just put the plugs back in again at the back. Can you hear me now?
Yeah, I think it's much better now.
Okay, I think maybe it was the sound because I think connector must have got knocked. Because I can hear you okay, you're not breaking, but it's me. Let me right, let's have a go. If it breaks up again, then we may have a half an hour break and we'll see if we can get it sorted. But let's carry on. Let's share screen again. Again, many apologies for this. Okay, yeah, so we want to know what the conformation of an antibody is like as well, and to make sure that the conformation hasn't been disturbed by storage or whatever conditions are used in the process of manufacturing the antibody and getting it to the patients. And also, if you make changes like adding on pegylated groups or changing glycoforms, making sure that the conformation is not compromised, because don't forget, the antibody has to bind and cross-link antigens and also bind to the effector as well. And if things are all compacted up or the conformation's been distorted, then that might be affected. So I'm going to show you now how we assess the conformation. It's more difficult than, or difficult to follow compared to the measurement of purity. I think that was quite straightforward to follow. But I'll take you through the steps so you can see how we can assess the conformation of an antibody molecule and some of the complications that we have to deal with. And we're talking about solutions now, not the crystal structures of antibodies. If we're representing conformations in solution, we generally use models. And if it's a globular protein, like lysozyme, ribonuclease, or even the Fab and FC fragments, the individual fragments of antibody molecules, then we can use simple models. So crystal structures, as I said before, are difficult to obtain for antibodies because of the flexibility of the hinge regions. And that means, although you can crystallize antibodies, it's very difficult to interpret the electron density maps because they're not constant because of the flexibility, making it very difficult for us to get structures by this technique. They're also very big molecules. They're 150,000 or so in molecular weight, so they're too large for techniques like nuclear magnetic resonance to obtain 3D structures about. But what we can do is we can measure what's called the solution properties or hydrodynamic properties of the molecule. We can also measure the hydrodynamic properties of the individual domains, the FC and Fab domains, and we can also get the crystal structures of the Fab and FC domains. It's very difficult for the intact antibody molecule, and generally speaking, we can only do that for antibodies which have got no hinge or very little hinge region. But what we can do, we can get crystal structures of the individual Fab and FC domains. And the idea is we can combine the solution measurements on the whole antibody molecule with knowledge from the solution measurements of the individual domains, the Fab and FC domains, and then combine that with the known crystal structures of the Fab and FC domains to find out what the structure of the intact antibody molecule is. So we combine three pieces of information together: the known crystal structures of the Fab and FC domains, the measured solution properties of the Fab and FC domains, the measured solution properties of the intact molecule, and then combine these three bits of information together, and we can predict what the structure of the intact antibody molecule is. Not in terms of the detailed atom by atom resolution, but in terms of the orientation of the domains, i.e., find out whether we've got a compact antibody, where the domains are closely folded in, or whether they are stretched out. And this process of combining solution or hydrodynamic, that's what hydrodynamic is, it means water movement or solution measurements with crystal structures of the domains together, we call this crystallohydrodynamics, combining crystallography or the limited crystallography information we have on antibodies with hydrodynamic data. So using crystallohydrodynamics to work out the orientation or conformation of the antibody. And let's show you how we do it. Right, now when we're representing conformations of proteins in solution, we can use simple shapes to represent the overall conformation, and these are called ellipsoids. Ellipsoid is like a three-dimensional ellipse or a squashed sphere, and this ellipsoid has three axes or semi-axes. It's got a long axis A, a shorter axis B, and an even shorter axis C. And when these axes are all equal, A equals B equals C, you have a sphere. When they're not, you have an ellipsoid. And the shape of this ellipsoid is determined by these two axial ratios, the ratio of that one to that one, and the ratio of that one to that one. So the shape is represented by the axial ratios A over B and B over C. And when these axial ratios are 1, you've got a sphere. When they're not 1, you have an ellipsoid. Now, unfortunately, we can't represent an antibody by this shape because they're very asymmetric structures. You know, they have these different arms which are not necessarily planar. So these simple ellipsoids, which we use for globular proteins to represent the shapes in solution, won't do. What we can do instead is we can use what's called bead models, where we approximate the conformation of a complex molecule. This is not an antibody molecule, it's just a schematic complex molecule, which looks rather like, what does that remind you of? It's rather like a snail. Yeah. So we've got a snail-like molecule we're representing by these spherical beads. 1, 2, 3, 4, 5, 6, 7, 8. There's these 8 beads of different sizes, represents the conformation of this complex molecule. And what we can do is we can model the arrangement of these beads, and then we can use computer programs called Hydro and Solpro, developed by our colleague Garcia de la Torre in Murcia in Spain. And so for a given shape, we can work out what the sedimentation coefficient is. We know the mass, of course, as well. And the idea is that you measure your sedimentation coefficient, first of all, of your complex molecule using sedimentation velocity in the centrifuge. You measure the sedimentation coefficient, and then you create a model of your protein. And then using this program's Hydro and Solpro, from how these beads are arranged, you can work out what the sedimentation coefficient of that molecule should be. So you work out what the sedimentation coefficient of this model is using Hydro and Solpro, and then compare that with the measured sedimentation coefficient, which you measure using the ultracentrifuge, and then, oh gosh, they're not the same. But then you keep on changing this model until you get agreement with the sedimentation coefficient which you measured in an experiment. And then if you get agreement, then this might be the correct model. I say might be. So by modeling the molecule in terms of these beads, finding out what the sedimentation coefficient of this model is using these programs Hydro, Solpro, comparing it with the actually measured value, and then just changing, refining the model until you get agreement. This is called model fitting or modeling the conformation of the antibody molecule. And there's an even more refined method we can use. Instead of this conventional bead model, we can just represent the surface of the molecule, this complex snail-like molecule. And by using lots of little mini beads, we can get a better representation of the surface of the molecule. And this is called bead shell modeling because you're representing the shell or outer surface of the molecule. And we can use Hydro and Solpro with this bead shell model. Right, and here's an example done, goodness me, 31 years ago now. This was the first demonstration that the antibody molecule IgE had a bent or cusp shape. This is the Fc domain here, represented by these beads. And these are the two Fab domains represented by these beads. So you produce your model, you obtain the sedimentation coefficient and this other parameter, which you can get from X-ray scattering on solutions of the antibody molecule. This is called the radius of gyration. So you create your model, you find out what the sedimentation coefficient and other parameters are based on that model. You compare it with the experimental data, and then you adjust the model until you end up with agreement. And using this type of modeling, back in 1990, we were the first ones actually to show that the antibody IgE had a cusp shape in solution. It was a bit more complicated than that. We had to do some comparisons with this hingeless mutant antibody molecule called IgG-MCG. But that's basically how we did it, by model fitting the sedimentation data. And then later on, the NMR measurements were done on the fragments, and then X-ray crystallography became available on IgE. Because it doesn't have a hinge region, they were able to get the crystal structure of the IgE molecule out. And this confirmed the structure that we got 10, 15 years earlier using hydrodynamics and this bead modeling approach. And this agrees very nicely with the functional shape of the molecule. I'll just twist this around a bit. There we are. And you can see that our bead model agrees quite nicely with the pictorial representation of the IgE based on crystallography and the interactions of the constant domains with the high affinity receptor on the cell the cell surface. That's good. Right, but that was 30 odd years ago, and now we use these more refined bead shell models where we represent the surface of the complex molecule by this. By the way, there is no molecule that's shaped like a snail. I've simply showed this shape just to illustrate the use of this type of modeling to represent the conformations of complex molecules. So these days we tend to use bead shell modeling because it gives a better representation of the surface of the molecule. And here's some bead shell models. Right, now these are representations of the IgG2 and IgG4 antibody molecules, which are quite similar in conformation. So, as I said, we don't get atom by atom resolution with these methods. We get these overall shapes of the molecule, and the two Fab domains are here, represented by these ellipsoidal shapes, covered by these little bead shells. And then you have the FC domain here, again represented by this surface bead shell shape. In fact, these models look a bit like we have a type of sweet in the UK called licorice all sorts. Again, has anyone had any of these licorice all sorts before? Raise your hand, folks, if you had licorice all sorts. Nobody. These are very famous UK sweets made by Bassett, I think. And right. Actually, looking at the people, maybe the reason why you've not seen these is that there's no one from the UK. Am I the only one from the UK here, folks? Right. Okay. So take my word for it, folks. Where's George Blackburn? He can back me up on this. Or Victor. No. Okay. So. Tell you what, I'll type in the, I can't even pronounce it, licorice. Let me see if I can get it up. And screen here. Oh, 1 second, folks. Right. For some reason, it's come out. I've got this huge email address. That's not right. Let's go licorice all sorts again. Bear with me, folks. Right. I'll send you the Wikipedia page, licorice all sorts. Right, let's go back to Teams page. Okay, if you can click that link, folks, I've just given you. Actually, I can share screen. Probably easier actually if I share screen. This is the beauty of the online teaching now. We couldn't do this normally in a lecture, but let's see if I can share this screen with you. Yes. Right, can you see this screen of licorice all sorts?
Yes.
Yeah, we can see.
See that one there? The bead shell model. Looks a bit like an FC fragment. This could be your hinge region here. So that's a slight digression, folks. Let's get out of this now and go back to the main plot. Right, okay, so see the analogy there? That's what we call licorice all sorts models. These are bead shell models, like licorice all sorts. So these are models that have been created in the way I said. We've basically combined solution measurements we've done on the FC and Fab fragments of IgG antibodies, and we've done solution measurement of the sedimentation coefficient on the intact antibody molecule, and we've combined that with information from the known crystal structures of the FC and Fab domains, combined all this information together, and we end up with a sedimentation conformation parameter known as P, which stands for Perrin function, P. Now, this conformation parameter P is very useful because when P has a value of 1, it means that we have a sphere. And if it's greater than 1, it means we have something which is departing from a sphere. So what we can do is we can, from our measured sedimentation coefficient on the antibody molecule, and we can work out what the P value for our antibody molecule is, and we get these conformation P values for IgG2 of 1.22 and IgG4 it's 1.23, very close. So IgG2 and IgG4 are virtually indistinguishable in terms of the hydrodynamic properties. So we have these experimental P values we get from the sedimentation coefficient of the antibody molecule, and also we've used information from the crystal structures of the domains and the sedimentation properties of the domains to get these P values. So they're not spheres, obviously, because they have a value greater than 1. But what we can do is we can create models of the arrangement of the FC and Fab domains and use those programs I mentioned before, which were Hydro and Solpro. So we create our bead models, these mini beads, based around the FC, the crystal structures of the FC and Fab domains, and then we piece the Fab domains and FC domains together, and we work out what the predicted p-value is using Hydro or Solpro, and we just change the angle between the domains. We just alter the angle between the domains until we get agreement with the experimental p-values. Again, this is called model fitting, adjusting the model so that the predicted values for a parameter, in this case, the shape function known as p, which comes from sedimentation coefficients, and get that to agree with the experimental values. And what happens is that these open conformations are consistent with the experimental p-values found for these antibody molecules. And then we can go even further, because with IgG1, which has a measurable hinge region, we can again represent the hinge region by these bead shells. And then for IgG1 with the hinge, we find experimentally the value is 1.26, and this agrees again with these open conformations for the p-value. P-values which we get from measurements of the sedimentation coefficient properties of the molecule. To repeat, by comparing the experimentally measured values with the model values, we can adjust the models which give the best agreement with the experimental data. So that's how we get the confirmation. That's how we can check whether a molecule, after storage or after some sort of manipulation, changing of an amino acid or whatever, has had an effect on the conformation of the antibody molecule. We can use this solution modeling of this type. But there is a slight problem with this type of modeling. Can anyone identify what this problem might be? Okay, well, I'll tell you. The problem is that you can find some orientation of the domains which gives agreement with the experimental value. That doesn't mean the confirmation is that. All it means is that you have agreement between your model value and the experimental value. However, there could be other models which you haven't tested for, which give equal agreement to the experimental data. And we call this the uniqueness problem. And that means that your model is not unique, or not necessarily unique, to the experimental value which is being modeled. So what we do in practice is that we don't rely on a single experimental technique like the sedimentation coefficient, but we also model other hydrodynamic data. And the biotech students, don't worry about this, because you won't meet this elsewhere in your courses. But just be aware that there are other hydrodynamic data which we can model in addition to the sedimentation coefficient to get around this uniqueness problem. Based on X-ray scattering, I mentioned that before, this is the RG parameter, and also based on the viscosity properties of the solutions of the antibody molecules. So just to repeat again, although a particular model may give a confirmation parameter P, which you get from sedimentation, in good agreement with the ultracentrifuge data, there may be other models which also give good agreement. This is called the uniqueness problem. And to deal with this, we need other hydrodynamic data. And I mentioned the radius of duration from X-ray scattering of solutions and the intrinsic viscosity parameter. And this is the subject of ongoing research we have in the NCMH, which is our laboratories, in collaboration with scientists like Roy Jeffress, who you met last week, and also colleagues from industry who you'll meet in 2 or 3 weeks' time when we have the lectures from the people at AstraZeneca, Sanofi, Arecor, and GlaxoSmithKline. So that's how we get the confirmation of antibodies. It's a bit more complicated compared to purity, where we're just measuring the sedimentation coefficient distribution. With confirmation, we have to do modeling, and that means use of computer programs, the creation of models using those beads, those complicated beads, because we can't use simple ellipsoid models, which are suitable for globular proteins, but instead we have to use the complicated bead modeling approaches, using those programs, Hydro and Solpro, working with this confirmation parameter known as P, which we get from or relates to sedimentation. And then to get around the uniqueness problem, we should really be comparing with other hydrodynamic data as well. But that's beyond what you are required to know for this course. But if you are interested, then you can look at these references here, which I've given you. This is one on the purity. This is in the Journal of Pharmaceutical Science, and I think I put that on the Moodle for you to access. There's the IgE one. We're modeling the IgE conformation. That was published, goodness me, 30 years ago in Biochemical Society Transactions. And the basis of the crystal hydrodynamics strategy for working out conformations in solution outlined in these publications here. This one is that's with colleagues from what's now AstraZeneca in Cambridge. Yeah, okay. These are on the Moodle, so if you're interested, then you can look at these publications. I'll look at this one, but this is should be easier to follow because it's based on the purity. And if you want a challenge, you can look at these papers here. Okay, so that in a nutshell is how we use solution properties of antibodies to investigate the purity, freedom from aggregation, and the conformational integrity, centered around the analytical ultracentrifuge instrument. There we go. Great, so that's 1 minute to 4. I think that brings us to a close. Again, it's one of those lectures where you're going to have to go away and absorb some of the material. I think after you've been through it once or twice, you should be able to get a good understanding of how we assess the properties of antibodies. Right. Last week it was Asda that were coming in. This week it's my one of my granddaughters. You can hear that. I'll just 1 second, folks.
Sorry, I think you're muted.
Sorry again. Sorry, Anastasia. Has anyone got any questions you want to ask me now, or would you rather go away and absorb and ask me when you've had time to think through what we've just gone through?
I don't really have any questions.
Okay. I mean, when you do the assignments, there will be a question. I think it's an optional question. I think you don't have to answer. There's a question on the use of the ultracentrifuge for purity and confirmation. But I'll be officially setting those questions next week. We're just finalising them now, actually, with Natalie and 1 or 2 others. But there will be a question on the solution properties when you do the assignments, which will be optional. But yeah, I would play those videos back I gave you again, and then go through this lecture again after I've finished and put the recording on YouTube for you. And now we know what a licorice ore sort is. Yes. Yeah, it may sound a bit silly, but sometimes when you can identify something which appears quite complex with an everyday situation, like the shape of a particular sweet or something, then it does help you to understand that concept a little bit better, and make that concept sound a little less fearsome than what it actually really is. Okay, so next week it'll be Manny talking about the pegylation of antibodies, which is a very clever way of increasing the stability and other things of antibody preparations. And we'll be also hearing from Dr. Duncan Hannant, who's a veterinary antibody expert and does work with the horses, particularly the National Centre for Animal Health at Newmarket in Cambridgeshire. And he'll be talking about therapeutic antibodies. So it should be a good week again next week. And then after that, we've got the guys coming in from industry, which is also very, very interesting. So I think it's going okay so far. All right, folks. So I'll say goodbye then, and wish you a nice week. Looks a bit weather-wise. Got say goodbye to all the snow and things. Looks like we're entering spring. Looking outside anyway. So and also the reason for optimism is the fact that the infection rates and all this sort of stuff with the virus is now falling dramatically. So hopefully normality will return, or near normality will return in the near future. So that's something to be pleased about. Okay, folks, see you next week. Bye.
Thank you.
Thank you. Bye.
Bye.
Bye.